Obviously, today, DNA plays a central role in our understanding of genetics, and it's easy for us to talk about, haploid sets of chromosomes fusing to form an embryo, and then that one allele on one chromosome, which has a specific nucleotide at one spot, then causes this change in a protein, etc., which then affects phenotype. So this is central to our modern understanding of biology. But obviously this wasn't always understood. One of the greatest weaknesses in Charles Darwin's uh, um, Origin of Species was he knew inheritance and variation were key, but he couldn't explain them. And this is why, in genetics, we give such attention to Mendel, because for thousands of years, no one understood heredity, and he, in his garden, figured it out. But come the 20th century, a number of great advances were made. In 1928, Griffith showed that there was a hereditary molecule, which was a big advance because, back in the day, a lot of people just assumed that there were other forces at work. So, for example, a pregnant woman's state of mind or what images she saw could actually then determine a trait in her fetus. Um, and the way that Griffith did this was through the transformation experiments that I had previously described. So in transformation, if, say, this cell dies on the right, the cell on the left, if it's a bacterial cell, can use its biomolecules as food. It can then break down the carbohydrates, proteins, nucleic acids, etc. But on rare occasions, the second cell actually takes up the DNA from the dead cell, recombines it with its own host chromosome, and now it could then have a new trait, which was gotten from the DNA from the dead cell. Well, Griffith used variations of this experiment to show that there were, in fact, hereditary molecules, that there was a physical something which was going from one cell to the next. Now, later, Griffith did show that this was DNA because you could digest RNA and protein and other molecules, and the experiment still worked. But when you digested DNA, it stopped. So even Griffith showed that it was DNA, but no one really believed him. And the reason was DNA is a simple molecule. It has the same double helix shape. It has only 4 nucleotides composing it. While in contrast, proteins are far more complex. They have more building blocks. There are 20 amino acids. There's a greater complexity in structure. And people with a little bit of hubris said, oh, look at me, I'm so complicated. How could that simple molecule DNA be the one that codes for an organism as complex as me? It must be protein, which is the hereditary molecule. But that was conclusively shown not to be the case in the Hershey-Chase experiments, where virus was used. Now, viruses, they can have a couple different kinds of nucleic acid. The virus used in this experiment had DNA. And so viruses, the ones which lack an envelope, have only 2 parts. The DNA is the hereditary molecule, and there's an outer protein capsid. So it was known that the virus must be injecting something into cells, which then caused the cell to manufacture virus. And so there were only 2 possibilities. Was it the DNA which is injected, or the protein? And so what they had done is they had different strains which were allowed to be formed with different radioactive elements. And so they had one strain with radioactive DNA and another strain with radioactive protein. And then what they could show by allowing the infection to start, then hitting the setting on the blender, and then disposing of the wastewater, that it was the radioactive DNA which was inside the cell, and the DNA radioactive protein which was in the waste water. This video that I'm making now is just a quick overview of this playlist. One of the things that I do with my students is kind of walk through the steps of these two experiments because they were so important. I use zombies instead of the original viruses or the mouse strains with pneumonia. And so if you were having trouble with the idea of, either of these experiments, either the Griffith experiment, which demonstrated that there was a hereditary molecule, or with the Hershey-Chase experiment, which demonstrated that the hereditary molecule was DNA, then you might want to do these. Once again, I tried to walk through the same steps of the experiment, but just, obviously using more humorous zombies than the original setting. So that's at the bottom of this playlist. Another key discovery which came early on was the observation that the amount of adenine always equals the amount of thymine in DNA in the nucleus. And then the amount of cytosine equals the amount of guanine. Now we know why, as I'll be pursuing presently. It's because in the double helix, adenine binds to thymine, so every adenine has a thymine partner, and every guanine has a cytosine partner. But that was not known at the time, and it was important then to establish that fact. Rosalind Franklin was the one who took the photographs, which would be key in understanding the double helix structure of DNA. Unfortunately, because of her untimely passing, she could not be given a Nobel Prize for her work, and her role was not fully appreciated until after her death, and probably at least part of that was due to women not having the prominent role that they deserved in scientific discussions and the like. And once again, her work was not appreciated until later. Watson and Crick, using those photos and the other data of the day, they then determined what the structure of DNA would be, because if you had pictures like that and you could then measure, the pictures and you knew the components, there were only so many ways, that these parts could be fit together. And then obviously, given the dimensions, some of the ways didn't work. So that we see that DNA is a double helix. And in this double helix composed of nucleotides, which I'll be describing presently, the phosphates and the sugars formed the outside. So if you twisted it so that it stood straight, that these would kind of be the uprights on the ladder, and that the nitrogenous bases would then be the rungs of the ladder, and that there were two strands of DNA that ran in opposite directions. They were what's called anti-parallel, as I'll be describing once again in a second. One went 5' to 3', the other one went 3' to 5', and that there was complementary base pairing where adenines bound with thymines, etc. Okay, so I'll go through all of that presently, but this then revolutionized our understanding of biology. So there is a genetic code, a physical molecule. So for thousands of years, a lot of things were attributed to more mystical explanations, but now there is a code for life. In fact, it was so powerful. If you were to go into one of my cheek cells and take out a nucleus and put it into an ovum, I could have a twin, and the same with you. There is a code for you in the nuclei of your cells. And if you could put that into an ovum, then now a cell could develop following the same code as you once did, and develop your hair, your eyes, your blood type, etc. There are more than 3 billion nucleotides in this code, and this code is then made of these double helixes, where one strand's thymines bind to the other strands, adenines one strand, cytosines bind to the other strands, guanines, etc. And there is information then here. All right, and so this particularly is the sequence for the gene for beta hemoglobin. And so cells know how to make beta hemoglobin by reading the DNA code. We now know that the DNA code is in the nucleus with a little in the mitochondria. It is then copied and proteins are then made in the cytoplasm. So then just to review a couple of the points made there with a little more detail. DNA nucleotides have these three parts, as we'll see. There's a phosphate here in orange, there's a pentose sugar, and then here there are nitrogenous bases, so-called because they have nitrogen in them, as you'll see. There are 4 nitrogenous bases in DNA. There is guanine, cytosine, adenine, and thymine. And each of these then forms a partner with a specific complementary base pair. So thymine will bind to adenine, forming 2 hydrogen bonds. Adenine will bind to thymine, forming 2 hydrogen bonds. Cytosine will bind to guanine, and guanine will bind to cytosine. And so the sequence of nucleotides on one strand then matches the sequence on the other strand. It is often important, the distinction between the 3 hydrogen bonds which unite the guanines and cytosines and the 2 which unite the adenines and thymines. When we talk about promoters of genes, where genes start, or origins of replication, we often see that there are sequences like the Tata box, etc., which have lots of adenines and thymines. The reason this is significant is it's easier to start to separate a double helix where there are only 2 hydrogen bonds holding these together. And so this is significant, and very often, as we'll see later, the adenines and thymines are easier to pull apart, and so those regions can then signify portions where the double helix gets unzipped. Okay. So I repeat that here in this video, which then just has this more 3-dimensional structure. So a couple of things. Note that there is nitrogen in the nitrogenous bases. The nitrogen atoms are shaded pink here, and that adenine and thymine are oriented so that there can be 2 hydrogen bonds which form between them, while guanine and cytosine, there are 3. And because of those 2, as I have mentioned, here are promoters where genes start. And notice the sequence, you know, it's all thymines and adenines. Why? Because it is easier to unzip these areas with the 2 the 2 hydrogen bonds at each spot. So here you can see guanine and cytosine in contrast. When they align, there are 3 areas where hydrogen bonds, which, you know, once again, going just back to chemistry, then are the attraction between the oxygens, which have a partial negative charge, oxygen being very good at attracting other electrons. Electrons tend to spend a little more time around oxygen atoms. And so the slightly positive hydrogen pole and the slightly negative oxygen pole, they are then attracted to each other, forming hydrogen bonds. And the hydrogen bonds then help to keep the double helix then together with 3 hydrogen bonds between the guanines and cytosines, and 2 between the adenines and thymines. Now, at this point, we should probably label the nucleotides because we need to refer to the specific order of the carbons. So once again, in this animation, the round dot is a phosphate group. The pentose sugar is here, and then this is the nitrogenous base. Now the nitrogenous bases can either have two rings, in which case they're called purines, like adenine and guanine, or they can have one single ring, in which case they're called pyrimidines, like thymine, cytosine, and uracil. So in this pentose sugar, they're called pentose sugars because there are 5 carbons here. We can number the carbons, if there are 5. So the number 1 carbon is the one that attaches to the nitrogenous base. The number 2 is important in determining whether it's deoxyribose in DNA or ribose and RNA, as we will see presently. The number 3 is important. That's pointing downwards to where the next nucleotides phosphate group will attach. Here it would then be the number 4, and then partway on this chain would be the number 5. So as such then, this nucleotide, we can indicate its directions. We could say here's the 5' end up top, here's the 3' end on the bottom. So that's important. And when complementary bases form, they form in this way. When we look at how those hydrogen bonds line up, that if this nucleotide has its 5' end pointing up and the 3' end pointing down, then its partner then has to have the opposite orientation, and has to be what's called anti-parallel. So the opposite one has to run 3' to 5'. So in that double helix, the 2 strands of DNA are anti-parallel. One runs 5' to 3', the other one runs 3' to 5'. So I pointed out that number 2 carbon. And so let's now compare ribose and deoxyribose. So once again, nucleotides have 3 components: a phosphate group, which is a phosphorus atom, and 4 oxygens. And so RNA nucleotides and DNA nucleotides, they have the same phosphate groups. There are nitrogenous bases, so called because there's nitrogen in them. The nitrogen atoms have been painted, have been colored pink here. And there is no difference between, say, the adenine of an RNA nucleotide and the adenine of a DNA nucleotide, or the guanine of an RNA nucleotide and the guanine of a DNA nucleotide. So this is adenine. And so the nitrogenous base is identical when you're comparing these two. What varies between DNA and RNA is the sugar. Now there are pentose sugars in each case. So 1, 2, 3, 4, 5, 1, 2, 3, 4, 5 pentose sugars in each case. But if you notice, if this is the first carbon, then this is the second. And notice in the second carbon here we have an OH group in what's called ribose. But here we have 2 hydrogens. So there's another hydrogen on the other side. You just can't see it. So both of them have 1 hydrogen. But here, instead of having an OH, it just has an H. So this one is missing an oxygen. So it's what's called deoxyribose. It's identical to ribose, but it's missing an oxygen. And so the nucleotides have 3 components, one of which is a pentose sugar. And it is the presence or absence of oxygen on the number 2 carbon, which determines whether this is the pentose sugar ribose or deoxyribose. And thus then this determines whether we're talking about DNA or RNA. And then the same would be true of cytosine. The phosphates are identical. The cytosines are identical to nitrogenous bases. It is the pentose sugar which either has the oxygen in ribose or lacks it in deoxyribose. In guanine, it would be the same, where the phosphates are the same, and the nitrogenous bases are the same. It's just the presence or absence of the oxygen. However, in the last nucleotide, there are 2 differences. So the phosphates are identical. The ribose has an oxygen, deoxyribose lacks an oxygen. But here, the nitrogenous base is different. DNA uses the nitrogenous base thymine to bind to adenine, while RNA uses the nitrogenous base uracil. So the 4 nucleotides used to elongate a DNA chain are adenine, cytosine, guanine, and thymine, while in RNA molecules, it's adenine, cytosine, guanine, and uracil. And so once again, we have a double-stranded DNA molecule where the phosphates and the sugars, they're on the outside. So if you think of the uprights of a ladder, while it is the nitrogenous bases making up the rungs of the ladder in the middle. The hydrogen bonds which form between the nitrogenous bases will only line up if the strands are anti-parallel. So if the nucleotides on the left are running 5' to 3', 5' being the one on top, then the nucleotides on the right will run 3' to 5', with the 5' on the bottom. Now, I'm going to then in the next video go through DNA replication. And for my students, there'll be an exercise where they drag the numbers to the various atoms. So, that's the number 1 carbon, the number 2 carbon, etc. And then you'll get to distinguish between ribose and deoxyribose. So here in deoxyribose, there is not an oxygen there. You can then look at 2 sugars and identify which one is the ribose, which has the oxygen, which one is the deoxyribose, which lacks the oxygen. So thus this would be the one that is in RNA, etc. There are, once again, 3 parts of a nucleotide. The phosphate and the pentose sugar, they make up the rungs on the outside of the ladder, while it is the nitrogenous bases, they make up the uprights, while the nitrogenous bases link in the center, forming more or less the rungs. And so you can see here in pink, these are nitrogen atoms present here. Once again, those nitrogenous bases, they come in 2 forms. If there is a double ring, like adenine and guanine, then we call it a purine. While if there is a single ring, like cytosine, thymine, and the uracil we find in RNA, it's what's called a pyrimidine. So in those pairs, we find a purine binds to a pyrimidine. And that was part of what Watson and Crick determined, because if you had 2 pyrimidines joining, then DNA would be too narrow, more narrow than what the measurements were. If 2 purines joined, then it would be too wide. And so only if a purine bound to a pyrimidine did the length fit what it was measured to be. So it is the 5' carbon which binds to the phosphate. This 3' carbon is going to bind to the phosphate of the next nucleotide. And then if this is a thymine, then adenine would be the complementary base, just trying to get to the last one. And so if this here were adenine and you were asked, what base is complementary to it, only thymine then would be able to form the hydrogen bonds, which would then allow that. Now notice that here, the phosphate is pointing up. That is the 5' end. Whereas in the guanine, which will bind to it, the phosphate is pointing down. So while this one is running 5' to 3', this one here is running 3' to 5'. Okay, and so this is the code of life. All right, so we live in an age of biotechnology where one can engineer life. One can now change DNA. or insert DNA molecules from one source to another. So there is information which can be coded here, and ultimately, if the DNA from one of your cells can then form a twin of yours, it is these molecules which then hold the their order holds the order, the instructions on how to make hair and livers and bones, etc. Just like the letters of a language can be arranged to have meaning, these nucleotides can then be arranged to have meaning. So this was a quick overview of the structure of DNA.